Okay, could somebody confirm you can hear me?
Yes.
Thank you. Is that all the further we got? I suppose. All right. Sorry for being late, but I had to verify that it was me who was logging into Zoom, and that always takes time. Let me close a few things down here. Come on. There we go. All right. Today is November 21st. And I think, yeah, this will be our only meeting for this week. That doesn't seem right. Oh, okay. I'm looking at the wrong day. Let me look again. Yeah, we're supposed to be starting chapter 14. We're not there yet. We're still on chapter 8, and we need to finish chapter 8. There will be no lab today. I will log in just for answering questions, but after about the first 15 minutes, I'll be logging out of the lab. I wonder, well, we'll go with it. Have a happy Thanksgiving. You should be working on your unknown project, and you can work on your unknown project during Thanksgiving week. Your unknown project is due Saturday, December 2nd, and your unknown notes pages will close on Friday, December 1st. Any question about any of that? All right, let's begin. So it looks like we've talked about negative selection via replica plating, and we ended here.
You're not sharing your screen.
Oh, thank you. Hopefully I'm recording. Yeah, I am recording. Whenever I'm talking about the dates and the schedule of classes and what we're doing, I mean to share my screen. I'll show you my syllabus there. So if you don't see that, then you can let me know right away. All right, let's go back to lesson 8. So genetic recombination is the exchange of genes between two DNA molecules. Oftentimes this happens in crossing over. This is when two DNA molecules actually come together, like the mother's chromosome A and the father's chromosome. We should actually call that A because they are the same chromosomes that come together in pair. So that would be like mother's chromosome X and father's chromosome X. And when they pair like that, they can cross over like this. And then the mother's chromosome had lowercase e, F, G, and H alleles, and the father had the capital alleles. We can then get the chromosome break right here, and then they switch parts so that this chromosome now has the mother's lowercase e, F, and G alleles or genes, and the father's capital H and capital I gene. So that's a recombinant chromosome. And then the other chromosome is a recombinant chromosome as well. So crossing over are when two DNA molecules come together, and yeah, they actually pair because that's the first chromosome, and that's the first chromosome, so they do pair. And then they cross over, and then they break and exchange pieces. Okay, that's what crossing over is. The physical manifestation of this, and that happens in meiosis all the time. I don't remember if it happens in mitosis. I'm not sure about mitosis, but in meiosis, I know it happens all the time. And it causes recombination. Now, recombination itself is a little more general than crossing over, because this is exactly what crossing over is. And whenever the genes move around, that's recombination. And recombination doesn't have to be an even switch like this. Like you could have the lowercase e gene move to the chromosome with the capital E, and then there will be two E alleles on that chromosome. That would be recombination also. Any question about any of that? So recombination and crossing over are frequently the same thing, but recombination is more general. It can be more than just crossing over. All right, let's talk about genetic transfer. Vertical gene transfer occurs during reproduction, like when two parents have their DNA come together to make the offspring. That would be vertical gene transfer. Between generations of cells, the parent cell will divide and then give its genes to the two daughter cells. Okay? And that would be gene transfer between generations, vertical gene transfer. Horizontal gene transfer is the transfer of genes between cells of the same generation, where DNA comes out of a donor cell and then it's taken up by another cell that we call the recipient cell. There are three types of horizontal gene transfer. There's transformation, conjugation, and transduction. Transformation and transduction do happen in human cells. The conjugation happens only in microbes. I'm going to talk about it only in prokaryotes, and I'm not certain if conjugation can happen between yeast, but it certainly does happen in bacteria. A transformation is where DNA comes out of a cell and then it exists as naked DNA in the environment. Usually the cell that it comes out of dies, but that's not always the case. And then DNA is taken up by a recipient cell. And you don't need to know, but the DNA taken up has to recombine with the chromosome. And then the DNA that combines with the chromosome can be expressed, and that can change the phenotype of this cell. Because now this cell has the lowercase a gene, and previously it never had that gene. That gene came from the DNA molecule coming out of a donor cell. Any question about any of that? Let me state that in bacteria, transformation happens about 10 times more frequently than spontaneous mutation. So when a cell picks up a new phenotype, as long as that phenotype exists in another cell somewhere, this cell can pick up that phenotype. And it's more likely to pick up that phenotype by transformation, in fact, 10 times more likely, than it is by spontaneous mutation. Any question about any of that? All right. So this is showing you that this cell picks up that molecule of DNA or a piece of DNA, engulfs it, gets digestion, indigestion, I guess, and then gets change phenotype from a gene or genes on that DNA. And there you can see the change phenotype. Any question about transformation? Transformation is important for bacteria because if a cell wants to pick up like a gene for antibiotic resistance, it is 10 times more likely to pick it up by transformation than it would be by spontaneous mutation. Conjugation is the exchange of genes between two cells. It happens in two ways. In the gram-negative cells, we have a sex pilus. Let me blow this slide up. Which is this structure of that cell there. And this cell has the structure, and we'll call that an F+ cell. And through that sex pilus, it actually can skewer an F- cell. That's a cell that is not F+ cell, and we'll talk more about that. But it has the sex pilus, and then it can actually use that sex pilus to winch the two cells together. They do not have to become this close because the sex pilus is hollow, and then a plasmid from this cell can move from this cell into the F- cell. And that's sexual conjugation in gram-negative cells. We'll talk a little bit more about this. Sexual conjugation can happen in gram-positive cells. And what happens is two gram-positive cells, usually of the same species, come together, and then they literally have their cell membranes fuse. I would say that the cell wall has to be removed in this region. And then the cell membrane fuses, and now the two cells are connected together, where the cell membrane goes like this. And the cytoplasm from this cell can move to that cell and vice versa, because they have a mating bridge right there. And when there's a mating bridge, that means a plasmid, small piece of DNA in this cell, can move into that cell or vice versa. And that is how sexual conjugation happens in gram-positive cells. Any question about any of that? All right, let's talk about sexual conjugation a little bit more in the gram-negative cells. We have an F+ cell, which has the sex pilus on it. And I already told you the sex pilus will go out and skewer an F- cell, and then literally act as a winch to bring the two cells together. They do not have to be this close together because that sex pilus is hollow, and the DNA can move from the F+ cell to the F- cell. The F+ cell has an F plasmid called the F factor here. You don't need to know that, but it does have a plasmid in it. And that plasmid can replicate so that there will be two copies of the plasmid. And that plasmid doesn't need to replicate when the chromosome is replicating. In fact, the plasmid will replicate when an F+ cell skewers an F- cell. And so that plasmid will replicate, and then one copy of the plasmid will move through the sex pilus into the F- cell. Now that F plasmid has a gene on it that codes for the production of the sex pilus. And so if we look at this cell here, it now has the plasmid, which is the F plasmid, which codes for the production of a sex pilus. So this cell here will grow a sex pilus, and we will no longer call it an F- cell because it has the F plasmid in it, and it will grow a sex pilus. So we call that an F plus cell. That will change the phenotype of this cell. Any question about that? And obviously it's going to change the phenotype because it's got the gene making the sex pilus. But any other DNA molecule on that plasmid will send that gene into this cell. Any question about any of that? All right. That's sexual conjugation. If you're wondering why they're calling it sexual conjugation, I suggest you ask your significant other. That's meant a little bit as a joke. The third way that horizontal gene transfer can happen is by transduction. Transduction involves a virus, and we haven't talked about viruses yet, or have we? Trying to remember. Let me check my syllabus. We have not talked about plasmid viruses yet, so let me introductory talk about viruses. And that is when a virus binds to a host cell. A host cell is a cell that the virus can replicate in, and when it combines with the host cell, the virus will contract and put its genome into the host cell. The plasmid portion, or not the plasmid, the protein portion of the virus stays on the outside of the cell, but the genome of the virus will come into the cell. And the first thing it will actually do is code for the destruction of the chromosome of the host cell. So it will secrete an enzyme. You don't need to know that, but it will break the chromosome into pieces. And the reason why it does that is the virus genome wants to call the shots in the cell that the virus infects. And the easiest way to call the shots in the cell is to destroy the host chromosome. And then the host chromosome isn't calling the shots anymore in the cell. It's only the genome of the virus, which is controlling what's happening in the cell. What the viral genome will do is make copies of itself and then go through transduction. I'm losing it. Replication, RNA transcription, and then protein translation. So it'll replicate the genome of the cells using the cell parts, and use the cell machinery to make viral proteins. And that will replicate the viruses. The viruses will then form, and the last thing the virus will do is code for the destruction of the cell. And when it destroys the cell, the newly replicated viruses will be released. Any question about that? Well, viruses produce a lot of offspring, and if they make a mistake, they're not going to worry about it. We can see a mistake being made in that there. Let me see if I can blow that up. And you can see the reddish purple is a copy of the viral genome. So that virus is correct. However, this is a purple genome, and that purple came from a piece of this cell's chromosome, and it mistakenly got put in the virus. Now, when this virus infects this cell, it will replicate not the viral genome into this cell. It will replicate the DNA or piece of DNA from this chromosome of this cell. And if this DNA recombines with the chromosome, then the chromosome will get a piece of DNA from this cell that can bring new DNA into this cell. And this cell's phenotype can change, and it'll change by the genes it gets from this cell. And you can imagine if this cell was antibiotic resistant and this cell was antibiotic sensitive, this cell could get from this DNA the gene for antibiotic resistance. Any question about any of that? All right. So the important point is that transduction, transformation, and sexual conjugation can bring a new DNA into a cell, which will then change the phenotype of this cell. And this will happen at 10 times the rate for either sexual conjugation, for a transformation, or for a transduction. Each of them happen 10 times more frequently than spontaneous mutation. So if this cell's phenotype changes, it is more likely to happen from transformation, sexual conjugation, or transduction than it is from spontaneous mutation. Any question about any of that? All right. We briefly talked about plasmids. Let me talk a little bit more about plasmids. They are a small, circular, extrachromosomal piece of DNA. So they are a piece of DNA, but they're not in the chromosome. And they are small, like this one only has, I don't know, 1, 2, maybe 3, 4, about 5 or 6 genes. And the chromosome has tens or hundreds of thousands of genes. So the plasmids are much smaller and they only contain a few genes. Plasmids are important to a cell because, especially a bacteria cell, because if genetic information is going to be exchanged between two bacteria, it is most likely going to be a plasmid that will be exchanged. And that always happens in the case of sexual conjugation in gram-negative cells, but it also happens in sexual conjugation of gram-positive cells. And it can happen with transduction, where a plasmid's put into the virus. And it can happen with transformation also. I'm not sure it happens 10 times more frequently with transformation than it does for chromosomal DNA. But like I said, a plasmid DNA is more likely to be taken in by this cell or to be exchanged in sexual conjugation or in transduction than a piece of chromosomal DNA. So plasmids are important in cells. You should know there are different categories of plasmids. You don't need to know the names of the categories or what they are. Any question about plasmids? If not, let's move on to transposons and insertion sequences. Transposons are segments of DNA that can move from one region of DNA to another. So if the transposon is on a human chromosome, let's just say human chromosome number 1, a transposon can move off of human chromosome number 1 and then move on to human chromosome number 2 or some other place in the genome of the cell. Because all it says is that they can move from one region of DNA to another, and it can be another chromosome. Transposons always have insertion sequences on their edges. Like right here, it's an inverted repeat is the insertion sequence. And it's thought that the inverted repeat helps the transposon move from one piece of DNA to another. Let me see if I can go back and show you that. Not really. So let's say a transposon moves from this chromosome to this chromosome, assuming, or I should say plasmid, from this plasmid to that plasmid, assuming both of these plasmids are in a cell. The transposon is a region of this plasmid and it's going to move to here. The inverted repeat helps the transposon come out of this plasmid and then insert itself into that plasmid. Come on, mouse. So that's what the inverted repeat does, or the insertion sequence. The transposon always has at least the transposase gene in the transposon. And simple transposons only have the transposase gene and the inverted repeats at the edges of it. Transposase is a gene that codes for an enzyme that actually cuts this piece of DNA out of the segment of DNA it's in, and then it then moves to another piece of DNA or region of DNA, and the transposase gene cuts that DNA, and then the inverted repeats help it fuse to the newly cut DNA. And then as it does, that makes the DNA connected, meaning that the inverted repeat will bind to this portion of the DNA. And it is true, it has to be ligated together. And then the transposase gene, and then the inverted repeat, which binds to this region of the DNA there. And yes, a ligase enzyme has to connect this part of DNA to that part of the DNA. So that's a simple complex, simple transposon, where all it has is the transposase gene and the insertion sequence, meaning the inverted repeats on the end. But a complex transposon contains other genes. Usually a complex transposon has a complete functional transposon on this end of the complex transposon, and then another transposon at this end. It's thought that the reason why that happens is that you need these inverted repeats on the end here, and rather than, I don't know, break the DNA here and then insert the extra piece of DNA inside, and you can't break the transposase gene, so it has to be exactly here or there. That if you just double it, it's just easier. And so that's why complex transposons usually have a functioning transposon over here and a functioning transposon over here, but the whole unit acts as one transposon. And then it can carry a piece of DNA from one region to another. And this can be important, like if kanamycin resistance is the gene in the complex transposon. Why that's important is that the transposon can bring the kanamycin gene into a plasmid, because transposons don't care where they move to. It can be from the chromosome to a plasmid, or a plasmid to another plasmid, or a plasmid to a chromosome. Any question about any of that? So the overall effect of a transposon is they shuffle up the genes in a cell. For example, if kanamycin resistance was right next to the gene for, I don't know, methicillin resistance, if this is on a transposon, it can move to another region of the DNA or to another chromosome. Any question about transposons? And there's a transposon which is actually moved into a plasmid. And like I said, this is important because plasmids are more likely to move from one cell to another cell. All right. If there's no further questions, let me end by talking a little bit about genes and evolution. Genetic variation, if it's new genes coming in to the population or the species, then the only way that that new gene can evolve is by spontaneous mutation, or by, I guess, chemically induced or radiation induced mutation. I didn't mean to say it's only by spontaneous mutation. It happens only by mutation. That's the better way to word it. And then crossing over, and the transposons can just switch the DNA around, causing new gene combinations. Crossing over and transposons do not directly change the genetic variation. They just sort of change the arrangement of DNA within a cell. But they do not bring a cell new genetic variation, as spontaneous mutation does. But once that existing genetic variation exists, it can spread by more than just mutation. It can also spread by transformation, conjugation, and transduction. These different forces working on genetic variation can drive evolution. And how evolution will proceed will be by natural selection, driving the evolution of the species in a direction to make the species more adaptable to their environment. Any question about any of that? All right. Any questions about Chapter 8? If not, let's go on to, I think it's Chapter 14. Oops, wrong one. Yep, Principles of Disease. Chapter 14, Principles of Disease and Epidemiology. In this slide, I'm showing you the major goals and a rough outline. Know the terms. I've only got 3 here. However, if you know the terms of this chapter, you will learn about half of the material in this chapter, meaning this chapter is very heavy on the terms. 2, understand the relationship of the normal microbiota and its host. 3, understand the stages of disease development. 4, understand what nosocomial infections are and how to control them. A nosocomial infection is also called a health care acquired infection. Any question about any of that? Or any question about what we're going to do? All right, not hearing anything. So let me proceed. Some terms you need to know: pathology, infection, and disease. Pathology is the study of disease. Etiology, the study of the cause of a disease. For example, the etiological agent of the disease anthrax is Bacillus anthracis. And we've talked about that before because this is the first disease where Dr. Koch showed that this disease was caused by a microbe. Any question about any of that? All right. Pathogenesis is the development of a disease. Infection is the colonization of the body by human pathogens or the presence of a microbe where they are not normally found. For example, all humans have E. coli in them on their mucous membranes of their gastrointestinal tract, especially of the intestine and the colon. If E. coli is found growing elsewhere in the body, it is an infection. So if E. coli is growing in the ear or in the lung, it's not supposed to be there. So that is an infection. Any question about any of that? Disease is an abnormal state in which the body is not functioning normally. What's the word I'm looking for? A communicable disease, or a disease which is infectious, is caused by a microbe. When we're saying that, you should understand that very few microorganisms are pathogenic. Some microbes are even beneficial to their host. So few microorganisms are pathogenic, only 2% or less. They have to have the genes to make them pathogenic. The rest of microbes are not pathogenic, and only some of them, a few, can cause damage to the host, resulting in disease. Any question about any of that? All right, let's talk a little bit about the normal microbiota and its relationship to its host. All animals and plants have large communities of microbes that are found in association with the living organism. These communities are known as the normal microbiota, the microbes that essentially, once they get established, tend to colonize the host and will live with the host for nearly all of their lifespan. Any question about any of that? The normal microbiota is only found in certain locations in the body, usually on the surfaces like the skin or on the surface of a mucous membrane. That would be like on the surface of the eye and the surface of the mouth. But they also live inside the body where the body cavities are, like the gastrointestinal tract, and the lower urethra also has normal microbiota. And once they get established, many of the normal microbiota live for most, if not all, of the host lifespan. That's most, if not all. Occasionally, when patients are very elderly, they can lose their normal microbiota. And usually what happens is a pathogen gets established and then grows so well that the pathogen excludes the normal microbiota. The disruption of the normal microbiota and the host balance appears to be associated with disease and impaired immune function. An example of that would be when a patient first starts taking an antibiotic. Assuming it's a gram-negative antibiotic, it will largely harm your normal microbiota. And that's because, what was the bacteria we talked about? Not the bacteria, the antibiotic we talked about. Was either streptomycin or, I don't remember. Anyways, if you get an antibiotic and you take it orally, you're exposing the bacteria in your guts to high concentrations of the antibiotic. And then the host cell will start taking up the antibiotic and then secreting into the blood for other cells to take on the antibiotic. And initially, in the patient, even after a vaccine, you will have very low numbers of the vaccine or the bacteria. However, later on, especially as the patient ages, their immune system will weaken. And then the normal microbiota may grow when it shouldn't grow, or it may not be able to keep out all human pathogens. The important part is disruption of the normal microbiota host balance appears to be associated with diseases and impaired immune function. For example, when you take antibiotics, if you get constipated afterwards, or you get diarrhea afterwards, that is probably a side effect of taking the antibiotics. And that's disrupting the normal microbiota host balance. And then the microbiota, especially the gram-negatives, become much fewer in numbers. And the host will be off, slightly sick, either with constipation or diarrhea or some other symptom, until the host microbiota balance reemerges. Doesn't have to be in the same location as the last one, but it has to establish a rebalance. And like I said, disease or impaired immune function can happen while the normal microbiota host balance is in play. Normally, when you take antibiotics, you get constipation at most for a day or so, and the same with diarrhea for a day or so. Sometimes I've had it just for a few hours. And that's because the normal microbiota quickly reestablish another balance. The human normal microbiota, and when we're saying the microbiota, I usually like to put the word human in there because different animals have different normal microbiota. For example, your kitty cat or your domesticated cat has a part of its normal microbiota, Bartonella henselae. And Bartonella henselae is not a part of our normal microbiota. And so if you get bitten or scratched by your cat, you can pick up Bartonella henselae because in the teeth or mouth and in the claws, Bartonella henselae tends to grow in the cat as a part of its normal microbiota. And Bartonella henselae in people is a human pathogen. It causes cat scratch fever, in case you're wondering. So the normal microbiota is found externally on the body, on the skin, the mucous membranes such as the nose, the throat, the eye, and the mouth. The gastrointestinal tract A lot of normal microbiota there. And then the lower reproductive systems, meaning the lower urethra. Example is places where the normal microbiota are not found include the internal tissues and organs. Tissues and structures will be the same. So tissue and organs, such as the blood, the cerebrospinal fluid, muscles, nerve tissue, and bone tissue. Any question about the normal microbiota? Now, the normal microbiota usually gets established for about the lifetime of the patient. But if you get some bacteria growing on you for shorter than that, and it's usually just a few days, a few weeks, or a few months, we don't call it the normal microbiota. We call it a transient microbiota. And the best example of a transient microbiota was when I was working at PCC in microbiology, and they had MSA plates, and I routinely cultured my skin to see if I ever had Staphylococcus aureus. And after years of doing this, I never had any Staphylococcus aureus grow on the mannitol salt agar plates. You don't need to know that. I'm just trying to tell you a little story about a transient microbe I had. And then I took my mother and I to visit my aunt, who happened to have a Staphylococcus aureus skin infection. And she was receiving treatment for it. So when she came over to hug me, I didn't think anything of it and said, oh, well, she's getting antibiotic for treatment of the Staphylococcus aureus, so it's probably not going to spread. But I then tested my skin about a week later, and sure enough, I had Staphylococcus aureus growing on my skin for the first time. And I said, geez, I must have got this from my aunt, and I hope I don't have this for the rest of my life. And the reason is that Staphylococcus aureus is a human pathogen. It frequently causes boils or a boil on your skin, but it can become worse and invade the in this case, it'd have to invade the store I was at and then invade a susceptible patient before it could get established in the patient. And then we'll talk a little bit more about this. It has to do damage to the patient before we'll then have a sign and symptom. The point is, I then tested myself the next term and the next term and for several terms after that. And I never saw Staphylococcus aureus ever again. And I breathed a sigh of relief saying, woo, good thing I got rid of Staphylococcus aureus. It was only a transient microbiota. It never became a part of my normal microbiota. Any question about any of that? All right. We've already mentioned, but let's go ahead and state it again. The human body has about 3 times 10 to the 13 human body cells. And I think this number is a little off. Let me check on that. I need to change that if it is. Wonder if I can open 2 versions of the file that have the same name. Come on. Of course, I can always rename it. I'll open that one. That'll have it. Trying to figure out how I move pages on this browser. Well, there we go. No, I've got the same number there. So unless it's, I didn't change it in that one, and I changed it in a previous term. What would be the previous term? The summer? Oop. Nope, it should have been changed there. So that number must be correct. I was thinking that number was wrong, but I only have it in one place here. And so I'm going to assume that number is correct. The point being is we have about 10 times, it's not exactly 10 times, so it is slightly different. I did change it. And that we have more than 10 times the bacteria, no, it's not a 13, 14. That is slightly more bacteria cells than we have human body cells. So they've actually done the count now. And the point is we have more bacteria cells than we have human body cells. And that begs to ask the question, are we more microbe than we are human? And don't let that freak you out too much. Because the answer is we have more microbes than we have human cells. Here's some normal microbiota in the nasal epithelial, in the stomach, and there you can see the bacteria in, it looks like a spirochete or something. And there's the normal microbiota, the orange cocci here. This right there and those right there are not bacteria. That's actually villi on the cells, human cells. The same with that. That's part of the stomach lining right there. And those aren't bacteria. That's actually sections of the cell that are outcropping. I don't know how else to word that. Here we have normal microbiota in the intestine, and you can see lots of different cells. That one almost looks like a cocci. That one is a coccobacillus. That one's a coccobacillus. That's a real short bacillus. That's a long bacillus. And that is food that's being digested. Any question about any of that? All right. We fairly recently discovered some normal microbiota in some surprising places. For example, in the last 10 or so years, we've discovered that the female bladder has some normal microbiota in it. The bladder was assumed to be sterile previously. And when we look at it, we find bacteria growing within the human bladder. And I've looked to see if anyone has ever done studies on males to find out if males have bacteria growing in their bladder. And I can't find any research that even looked at it. So I don't know the answer to that. But in the females, we definitely have normal microbiota that moved up and went inside the bladder against the flow of urine and potentially against the flow of gravity as well. We do know that the lower urethra has a lot of normal microbiota, and occasionally that normal microbiota can move up to upper in the urethra, meaning the upper urethra, and then rarely the normal microbiota can actually come up to the bladder, causing a UTI. And then it can also move from the bladder up the ureter, and then from the ureter to the kidney, in which case that's a very serious infection. So we always knew the bladder occasionally had bacteria in it, but we assumed it was from the results of an infection. And at least in females, we find bacteria within the bladder. We also find bacteria in mother's breast milk while it is in her breast. It was well known that the baby was getting bacteria when it was nursing, but people assumed it was getting it from the mother's external body, meaning on the outside of the nipple. But in reality, when they sampled mother's breast milk, they found bacteria inside the mother's breast. And then the third one, which I'm going to briefly talk about, is the placenta. They found a unique normal microbiota that is a human normal microbiota, and it only grows in the placenta, meaning there are some components of these microbes which are only found in the placenta. If you don't know, the placenta is something that separates the mother's blood from the fetal blood. And usually after birth, the placenta is born next. It's just a bunch of tissue that happens during pregnancy or birth, not birth, but what do you call that? Sexual recombination of the egg and the sperm, and then the fetal development. Why that they didn't expect a normal microbiota in the placenta is a surprise to me, because it's well known that the human reproductive system is open to the outside. And yeah, there's the placenta between the, what is it? Lost the name of that. The female organ that holds the baby. Whatever that's called.
The uterus?
Yeah, the uterus. Thank you. All there is between the uterus and the not the vagina, but the lower female reproductive system, the urethra, is the placenta, and not the placenta, it's a sphincter, but it's still open to the outside. Maybe it is the placenta.
Cervix?
Cervix, there we go. Thank you. I was trying to remember. My sister had cervical cancer and I couldn't remember the name of it. So thank you. Yeah, anyways, the cervix is open. And so I'm a little surprised that they didn't expect that the placenta would have normal microbiota, but it came as a surprise. And like I said, there are some unique microbes in the placenta that are only seen in humans when the female is pregnant, and these microbes are only found in the placenta. All right, let's talk about the relationship between the normal microbiota and the host. The normal microbiota can help the host. For one thing, the normal microbiota can engage in microbial antagonism with a human pathogen. Microbial antagonism is where the two compete for resources and space with each other. And if you have lots of normal microbiota, and then the human pathogen comes down, it may have no room to grow. Or maybe the normal microbiota have used up all the resources in the region, so then the human pathogen cannot grow, cannot reproduce, and will eventually die. So the normal microbiota can engage in microbial antagonism with human pathogens, such as preventing the growth by simply occupying the niche the pathogen is trying to occupy, competing for resources. The normal microbiota can also change the environment, making it less hospitable to the human pathogen. For example, the normal microbiota of our skin makes our skin more acidic. And that's also true with the normal microbiota in our lower urethra, making the environment more acidic. And that will prevent the growth of many human pathogens. Not all, but many. And then the normal microbiota can produce a bacteriocin, which is a protein that acts like an antibiotic. A bacteriocin is similar to an antibiotic, except it's much narrower in its range of function. For example, we will talk briefly that some antibiotics are narrow range and others are broad range. Broad means they can work against many different types of bacteria. And the narrow range means essentially the antibiotic only works against one type of bacteria, such as only working on gram-positive bacteria. Well, probiotics are much more narrow than that. For example, not did I say probiotics mean a bacteriocin works on other bacteria, but much more narrow than like one class of antibiotics, where you either only hit the gram-positive cells or the gram-negative. Like polymyxin B is an antibiotic that only hits gram-negative cells. Well, a bacteriocin produced by E. coli only inhibits Salmonella and Shigella, which are two human pathogens. It would not inhibit another normal microbe in the gut, such as a microbe which is a cocci, or a microbe which is gram-positive, because I mentioned that polymyxin B only works on gram-negatives. Well, in addition, the bacteriocin would not work on many gram-negative bacilli, even if it's killing off Shigella and Salmonella, like Enterococcus faecalis is a bacteria found in our gut. And in some people we have, trying to remember the name of it and I just lost, Lactobacillus lactis, Lactobacillus lactis and Lactococcus lactis. We can have these in our gut, and they're bacteria that help digest milk. And the bacteriocin would not prevent the growth of any of those cells, wouldn't harm those cells either. So a bacteriocin works like an antibiotic. And I often wondered why people weren't using a bacteriocin in place of an antibiotic. Because generally, if you know what the infection is, you want to use an antibiotic that works against the fewest number of bacteria possible, so that you minimize the side effects of the antibiotic. And I read a paper where bacteriocins were experimentally being used to treat against certain bacteria infections, such as Salmonella and Shigella. Any question about any of that? If not, let's mention that probiotics are the consumption of live microbes or the taking of live microbes. And the benefit is it's intended to exert some sort of beneficial effect on the patient. Like when you eat yogurt with live cultures in them, that can reduce the chance of getting a yeast infection. And that's well known. Any question about any of that? I guess we can also mention that sometimes when you take antibiotics, they will tell you to eat yogurt or take a probiotic, and that's to replace the bacteria you lost from your initial dose of antibiotics. All right, the normal microbiota and the host establish a relationship, and that relationship will be symbiosis, where the two different organisms or populations will live together. The normal microbiota and host are in symbiosis, but they're not just in any symbiosis. They're either in commensalistic symbiosis or mutualistic symbiosis. Mutualism is maybe the easiest to understand, and that's where the bacteria living in symbiosis with you or in part of your normal microbiota living with you. Mutualistic one would be where the organism, the microbe, gets a benefit, and usually they get a home and a food from us. Like the microbes in our stomach, they're given a home, and generally it's a good home because hopefully you're eating properly. And then the bacteria benefit us, like E. coli helps us to digest our food. So that's a mutualistic, symbiotic relationship of the normal microbiota. The other normal microbiota can form commensalism, where the symbiosis occurs, but it's commensalistic. One organism benefits and the other is unaffected in commensalism. So the organism, the microbe, benefits because it's given a home, it's given nutrients, but it does not help the host. It doesn't harm the host, but it doesn't help the host. Any question about any of that? All right, the third type of symbiosis is a parasitic symbiotic relationship. That's where one organism benefits and then the other organism is physically harmed. All human pathogens are parasitic, meaning they form parasitic symbiosis, and that's because they live on and in the host, but they also do damage to the host, and that's why we call it a parasitic relationship. Any question about any of that? All right, the normal microbiota does not normally cause disease, but occasionally it can. In which case, if the normal microbiota does cause disease, we call it an opportunistic microorganism, where some normal microbiota become pathogenic, meaning they grow and cause disease in the patient. Normally, the normal microbiota do not cause disease. In a healthy individual, their immune system will prevent the normal microbiota from ever getting established internally in the body. But occasionally, some normal microbiota can cause disease, in which case we call them opportunistic microbes. The individual may be very unhealthy, like they're about to die, and then they get an opportunistic infection where a species from their normal microbiota grows uncontrollably, and the patient has no immune system or a weakened immune system to keep the bacteria in check. Or if the patient is severely immunocompromised, like an AIDS patient, AIDS patients can get the normal microbiota to grow in regions where they should not be growing or to cause an infection. E. coli is normally found in the gut, and as long as it stays in the gut, it doesn't cause any problem. It's a part of our normal microbiota. But if it grows in the lungs or the ear, that's a place where E. coli should not grow. And here we call it a opportunistic pathogen, not an opportunistic microorganism, where it's growing where it should not, and then causing disease. And then Staphylococcus aureus is found in the nose of about 50% of Americans, but on the skin, nose and throat, I should say, but on the skin, only about 20% of Americans have Staphylococcus aureus on their skin. Staphylococcus aureus is a human pathogen, and it can cause boils and then invade deeper and get into the blood and then be spread, causing disease anywhere in the body. So Staphylococcus aureus can be opportunistic. Normally, in the nose and in the throat, it does not cause disease. But on the skin, it frequently is a human pathogen. And like I said, 20% of Americans have Staphylococcus aureus on their skin. Any question about any of that? All right, let's talk about Koch's postulates again. Remember, we're talking about the etiology of infectious disease, and the first organism that was ever shown, I should say the first microbe that was ever shown to cause disease was Bacillus anthracis, causing anthrax. And it was Dr. Koch who proved it, showed it happening. He then established Koch's postulates, which are used to prove that a given organism causes a given disease. This is, I believe, the second or the third time we've talked about this in this class. If ever we talk about something about 3 times in the class, you know it's important. And you should learn it because there will probably be a final question on it. Koch's postulate states that if we have a sick bunny and we take microbes from that bunny or animal and then grow them up, isolate them into pure cultures, grow them up, and then we inoculate a healthy bunny with a microbe that we think is causing the disease. And so it's one pure culture grown in vitro. And then we inject it into a healthy bunny. If the bunny becomes sick with this disease, that's implying that this organism caused that disease. And you can see the bunny is on its side here because the bunny isn't feeling too well. Same with that one there. That's a healthy bunny right there. That's why he's up in the normal bunny position. However, Koch said that's not all you need to do to show that this organism is causing disease. And he put into Koch's postulate, you must obtain the organism from the sick bunny that you injected into the bunny here, the healthy bunny, before it became sick. So you must be able to get that organism to grow in vitro and show that, yes, it is this organism that caused that bunny to become sick and the same organism that caused that bunny to become sick. And that's Koch's postulate. There is one other condition of Koch's postulate, and that is all organisms that have this disease have to have this organism causing the disease. Any question about any of that? Now, you should know that there are some exceptions to Koch's postulate. Such what as you do if you have microbes that cannot be cultured in vitro, like Bartonella henselae, we've tried for 100 years to grow that in culture. And we've never been able to get it to grow. We don't know what its growth factors are. We don't know what to supply the bacteria to get it to grow. And then another one is syphilis. This one they've tried to grow for over 100 years in tissue culture, and they've never figured out what they need to add to the culture to get the bacteria to grow. There's some growth factor that is being supplied by the host, in this case, the humans, and what it is, we just don't know. And so we've never been able to get a syphilis or Bartonella henselae that causes a cat scratch fever to grow in tissue culture. And if you can't get it to grow in tissue culture, you can't do Koch's postulates to prove that this organism is causing the disease. So how do we get this disease being caused by one organism? How do we interpret that? Another case is, what do you do if it's morally and unethical to give a human-only disease to a human? Like, for example, no one has ever injected the HIV virus into people to see if those people would then later on become AIDS patients. It's not been done for moral and ethical reasons. And yet we think HIV causes AIDS. And how do you think we know that? We can't show it by Koch's postulates. So how did we come about that? All right, I'm not hearing anything, so I'll go ahead and answer that question. And that is, we know that HIV is causing AIDS because they've studied very large populations of people, some of which, in fact, initially, the majority of which were HIV negative. And then they study these people over time. And some of the HIV negative people will become HIV positive people on their own, meaning we didn't inject it into them. And we then follow those people over time. And many of them were shown, which became HIV positive, were shown to later on develop AIDS. And those were the only people and the only organism that has been shown to cause AIDS, meaning they've tested other organisms that they found in people, and only HIV, that virus, is known to cause AIDS. Any question about any of that? All right, let's talk. Actually, we're out of time here, so I'll go ahead and end it, and we will start right here next time. Any questions before I go? All right, if you have questions, I'll be in the lab from 6:30 to 45. If there's no questions, you don't need to show up.